everyone, and today I'm here to celebrate, and we are going to do a get together. So if you are here and watching us on Facebook, please give a hi in the comments so that we know. So today is National DNA Day. So what's the importance of this day is that there was a scientific revolution that had happened few years back, and with that scientific event, we came to know everything about our own genome. Fantastic, right? So that was marked as April 25th on 2003 when Human Genome Project got completed. And from there on, this is DNA Day, celebrated as National DNA Day. So if you are here, I can see some people are here watching. Please say that you are here. All right. So as you know that generally I speak in this live, but as you know, I'm telling that it is a get together. So I will now call in my friends inside. So I welcome Dr. Udita Singh into the show. Hi Udita, how are you?
I'm all good.
Fantastic. So I think this for the viewers and also for you, Udita. That I think nobody can stop us from celebrating science. And there is a surprise guest whom I think if you see, all will be very happy. So let me just call in that guest and to my viewers, to all our viewers in the group. Uh, you can guess who is this. I think you know them. You know her. You guys know her. So yeah. Okay. We have our special kid with us. So that's Rajasiri. So today we three will be taking a few minutes of all of yours, and we will just try to celebrate National DNA Day. And that is a reason to hang out, basically. Nothing more than that, I guess. So, yeah, okay. So, if you guys are here, can you give me some hi? Can you give us, all of us, some hi, hello, so that we get motivated? Fine. So, in this today, what we are trying to do is uh, what we'll do is we will talk about different things. So first let me tell that I will talk about something very technical. That is, I will show you a fascinating device with which you can collect DNA from saliva. The show will be run by these two guys today. So okay, Udita, can you tell what you are speaking on today?
I'll be talking about the role of dental DNA, how the DNA is extracted, and how it is helpful to all of us as dentists to teach general psychology.
Fantastic, fantastic. So, okay, Rajeshri, what is that you will give us insights about?
Okay, so today we are going to celebrate National DNA Day on this platform. And I'll be talking to you a little more about DNA and a wonderful international scientific project that was carried out by various scientists across the globe called the Human Genome Project.
Fantastic. So I think Rajeshri will actually do the justice to today's National DNA Day by speaking about Human Genome Project. And I think I will let's start. And I think Udita, would you like to start?
Yes, ma'am.
Okay, fine. So it's to you now. Please go ahead.
Hello to all the viewers. Gargi ma'am for providing us such a valuable platform where people from specialty can come together and share our insights, what all we have learned. So to begin with, let me introduce myself first. I am Dr. Udita Singh. I am oral medicine and radiology specialist and a fellow member of Indian Board of Forensic Odontology. So today I'll be sharing my views on the various applications of DNA in the field of forensic odontology. So forensic odontology, in very layman terms, is a specialized branch wherein we apply the dental knowledge to establish the identity of the any person, any unknown person. So this word identity, it originated from a Latin word which means same or sameness, and it refers to the characteristics which makes us very unique. Like all of us are almost similar to each other, but still we have certain peculiar features by which one can differentiate one person from the another. And in this branch, we try to establish this identity by scientific means. So this, as all of us know, this proper and prompt identification of a person is very much required for both humanitarian and legal issues. The legal issues can be something like settlement of property issues or allowance of insurance claims, or in cases wherein to facilitate the marriage of existing spouse or for closure of case. In other cases, there might be cases wherein we want some scientific evidence of the age. Like in various dental institutions, daily basis we come across patients or young children who want to have a scientific evidence of their age. Like they want to play for certain tournament and they want to know scientifically whether they are 16 years of age or 18 years of age. So these are the various applications where this identity or age and all these are required. Apart from this identification is very becomes important in cases like natural disasters or mass disasters, in cases where are train accidents or aeroplane crashes. So these are the various and very short, few terms I have just tried to summarize what all applications can be wherein we want to have the identity of the patient. And this personal identification is essential to establish the right of identity of any person. Every individual has the right of identity, and by this we can ascertain that. Until date, there have been various methods to establish this identity. The traditional methods of establishing the identity of any person can be visual recognition, just by looking at the person or just by commenting on the personal belongings, like the type of clothes the person was wearing, the watch or shoes or belt. Just by the basis of personal belonging, we can know that this is the person who we or she are talking about. But this method is very robust method, and this is not a scientific method, and that's why it cannot be used in the court of law. And moreover, in cases like planned murders, the bodies can be decomposed or these personal belongings can be interchanged. So this is not at all a scientific method. So that is why we need something more scientific which can be used in the court of law to establish the identity of the patient. So these various methods, scientific methods of establishing the identity, can be broadly classified under two heads. So the first one will be the post-mortem comparative identification, and the second method will be post-mortem reconstructive identification. So the first method, which is post-mortem comparative identification, as the name says, for it to be successful, we need to have certain ante-mortem data. And with respect to dental field, ante-mortem data can be something like an X-ray which was taken at your dentist clinic, or a dental cast, or a photograph, or the case sheet profile that we maintain. But in various cases, these ante-mortem data are not available. Or, for example, suppose there is a mass disaster. In cases of mass disaster, these telephone lines are disrupted. Plus, it is not possible to retrieve all these data quickly. So all this delay in communication leads to failure of this post-mortem comparative identification. So what other method we have? The other method can be post-mortem reconstructive identification. In this, we try to reconstruct the identity of the patient. We try to say that this person might be a male or a female, looking at the skeletal differences, or we try to look at the specific dental features, like a person will have shoveling of teeth, presence of some accessory cusp, or some lip prints or palatal rugae pattern. Depending on that, we try to say that this might be the person about whom we are trying to evaluate. But again, the success of all these, all I have covered, this depends upon the nature and severity of the accident. Suppose there was a aeroplane crash where bodies they were burned, they were charred so badly that we cannot comment on these. So in all those cases where the bodies are decomposed or charred, we cannot do a soft tissue analysis. Then coming to second, which is presence of certain specific dental characteristics. Like in this, what happens? Suppose there is a closed population, there is a population where homogeneous population is involved. Suppose there is a train accident in a part of Uttar Pradesh. So all the people who were involved have almost similar features. If accessory cusp, talons cusp is a normal feature of that population, everyone will be having that. So again, this feature, based on this, we cannot comment upon the identity of the person. So when everything fails, the most efficient method of establishing the identity is through the DNA only. This DNA estimation is well established, it is scientific, it is running since ages. Like my partners will be talking to you about various other projects. So this is the most efficient method, and this has already been established in the court of law. And by doing this DNA examination, we can be 100% sure. We are not working on any guesswork. We are not just telling you this might be the case. Here we are very sure that this is the person whom we are talking about. We can have a very clear cut idea about the identity of the patient. We can talk about the gender of the person depending on various specific genes. We can tell the age of the person about whom we are talking. We can have an idea about the ancestry of the person. And so all this club together makes DNA a very efficient method. Then along with this, I'll try to bring to your notice about an Innocence Project. So this Innocence Project, this is a non-profit organization, legal organization. So this was found way back in year 1992 by at a law school. So the idea behind this project was, it is committed to exonerating the individuals who it claims were wrongfully convicted through the use of DNA testing only. So, if you go back and read about this Innocence Project, it includes the life stories of various people who have been sentenced to life imprisonment, who have been in the prison since long 20 years, 30 years. And why? Because they were, at a time, they were sentenced, they were proven guilty in the court of law, but when their cases were reopened and DNA testing was done, then it came to everyone's notice that the person who has been serving the life imprisonment was actually innocent. So this again emphasizes the importance of DNA testing, and it is not that all these cases were randomly done. Proper everything was done. Then DNA, all the skeleton characteristics were matched, dental features were matched, but still there was something, because again we cannot be 100% accurate if we are not using DNA in our protocol. So this is a one project which I was talking about. So this emphasizes how important it is to look in the DNA. And depending on the success of this, there is a web series also. This is broadcasted on Netflix by the name of Innocence series. So since all of us are more free during this quarantine time, so I will suggest all of you to go back and look at those Innocence series. In every episode, they'll tell you about life history of a life, how this person was convicted, what were the charges, how the court proceedings were done, and later how this case was reopened. And just by the introduction of DNA technology, these persons, they were wrongfully convicted. So now it has become mandatory. Whenever there are cases of bite marks and sexual assault cases, it is mandatory to collect a saliva so that we can extract DNA out of it. So in the next part of the session, Dr. Gargi ma'am will be telling you more in detail about how to collect saliva and all, what are the various tools that are available. So this is, again, I would like to stress, this is a platform which we can use to create more awareness, because it is important. Like, one person might be knowing one part of technology, and with such platforms, all of us can club together and do a more effective, more fruitful science. Okay, ma'am. With this, ma'am, I'll sum up.
Yeah, I think that was wonderful, Udita, and I think you have gave us a wonderful insight, and also whoever is watching with us, and I see many people are here with us, and that's fascinating to see, and I think that a Saturday night cannot be more fruitful the way we are hanging out. Yeah, so yeah, and that was wonderfully explained. Actually, even it just I just reminisced what I studied long back. And thank you, Udita, for this wonderful session. And I think now the platform goes to our youngest participant. Not participant, she is the host. Rather, I was wrong. Okay, so okay, Rajashree. So I think Rajashree will be telling us about the she will be actually the torch bearer of this particular day that is being celebrated globally, that is National DNA Day. And Human Genome Project is basically was a scientific revolution where scientists from all over the world came together, both private sectors and public sectors, that is government sectors, came together, and the intention was to bring what is inside our DNA, human DNA. And I think I will not speak today much on this, but I think Rajashree will give us more insights. And yes, again, guys, if you are here, please give us hi, because it's Saturday night, and we are speaking here. And I think when you guys tell hi's and hellos, then we feel very good. All right, so that's over to Rajashree, and I think she will take up the session for the next few minutes. Yeah, Rajashree.
Thank you, ma'am, for the wonderful. I'm Rajshri and I'm an undergraduate student, and I'm currently pursuing my undergraduation from NSVK Sri Venkateswara Dental College, Bangalore. So today is indeed a day to celebrate because it's the National DNA Day. I hope all of you have already heard about this wonder molecule called DNA. So let us go back and imagine purchasing a new phone. With the phone package, we do get a small booklet, and it's called the user's manual. But have you ever wondered that manufacturing the human body should also have a user's manual? Can you imagine that each cell in our body is equipped with this user's manual in the form of DNA? And this is known as the human genome, the user's manual to manufacturing the human body. So, scientists from all over the world, in an amazing effort to sequence this human genome, carried out the Human Genome Project. This was an international effort funded by the National Institutes of Health, and it was a 13-year-long project that lasted from 1990 to April 14th, 2003. So before going in detail into the Human Genome Project, I would like to tell you about the fascinating facts about human DNA. DNA is not like any other molecule. It has the information to generate the entire human body from a single cell. Can you imagine? So, as all of you know, there are 26 letters in the English alphabet. Can you guess how many letters make up the user's manual for this human body? Any guesses? There are only 4 letters, just 4, that make up the information in our entire body. And these 4 letters are A, C, G, and T. Adenine, guanine, cytosine, and thymine. So these 4 letters encode various genes and non-coding sequences within the DNA that help in manufacturing the entire human body. And the information of 3.2 billion of these letters, or 3.2 billion of base pairs, is present in each cell of the body. And each cell of our body is about 6 microns. Comparing that to a mustard seed, it is 1,000 times smaller than a mustard seed that we use in cooking. So that's how small the information to create the entire human body is present. That is, within 6 microns of space, the entire information is packaged, and a single cell has the ability and information to generate a complex organism like the human, consisting of more than 30 trillion, that is, or 37 trillion cells within one human body. For your information, a trillion contains about 13 zeros after 3. So those many human cells make up this human body, and each one of those cells has the information to generate the entire body. So now moving on in particular about the Human Genome Project. So as I told you before, the Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up the human DNA, and to identify all and map all the genes of the human genome from both a physical and a functional standpoint. The human genome not only consists of genes, that is, the parts of DNA that code for proteins, but also non-coding sequences. So this particular project, as I told you before, was a 13-year long project, and it started in 1990 and completed on the April 14th, 2003. An interesting fact here is that each cell in the body, as I told you before, contains 3.2 billion base pairs, and the funding for the Human Genome Project was again about $3 billion. So there is this coincidence here. And why would we like to sequence the human genome? What was the reason behind sequencing this? Here, the scientists wanted to record the patterns in DNA in every cell of our body, in every chromosome. They sequenced the pattern or recorded the pattern of ACGT in different permutations and combinations. And now, after sequencing the entire genome after a 13-year long effort, we are trying to correlate the different patterns of ACGT that correspond to a particular human phenotype or trait. So there are patterns within the human genome which correspond to your eye color, your height, your facial features, and so many of your phenotypic traits. So now it is possible to reconstruct using machine learning algorithms a face or an entire human body using the information that is sequenced through the human genome. And this sequencing is done through a method of gene annotation. And a pioneer in this field was Dr. Frederick Sanger. He was a Nobel laureate and a biochemist, a British biochemist, who first invented the Sanger sequencing machine. And using this, the first human genome was sequenced. So this is how we have started off. Human genome sequencing has a multitude of applications, and we are evolving and working on this field every day. And this has applications not only in the fields of genetics, but also in precision medicine, where we can give targeted therapeutics to diseases, dreaded diseases like cancer and cystic fibrosis, and several other genetically inherited disorders that can be specifically targeted and treated with modifying the genome and genes. So I find that DNA is a molecule that encompasses the intelligence that all of us humans collectively possess. Another interesting fact is that the entire genome, when sequenced, comes to about 2,700 pages of an encyclopedia book. You can imagine how that will be. Among these, all of us are similar except for just 500 pages. You can imagine how different all of us look, but that information to generate those phenotypic differences is condensed into just 500 pages of that code of ACGT combinations. So that is the fascinating fact about DNA, about the Human Genome Project, and about the wonderful work that many scientists have done, and their pioneering works are enabling us to generate better knowledge and generate better treatment modalities and make humanity one. I hope whatever I said was, made you a little more knowledgeable about DNA. I hope most of you will go back and study a little more about the wonders of DNA and the Human Genome Project. Thank you for this wonderful opportunity.
I think that was fantastic. What say, Udita? I think that was fantastic from Rajashree, and it was wonderfully explained and so nice, so nice. I mean, really amazing, amazing the way you told about Human Genome Project. So I think nothing simpler can be way of explaining how Rajashree gave us the insights. So, well. Now, guys, you know now about as Rajashree told many facts and facts about DNA and regarding and also regarding Human Genome Project, as well as Dr. Udita focused on the applications of dental DNA in forensics, or rather the way commonly we call as forensic odontology, and both ways where I will speak is about saliva. So my role is very small today because all these guys have done my job. I mean, I'm so happy. So, anyway, so I'll go straight to the point. And what I'll do today is to make things a little more interesting. I have some fascinating thing to show, not to speak much, but to show. So I will be, I don't know if you are able to see, so let me just try to, project this picture here. I'll just hold on in front of my camera. Um, I think you can see, guys. I think, is it visible? Okay, you see some device here. So I will be telling what is this. So I think this will be helpful. Like, for different purposes, as put on by Udita and Rajashree, that we need we need DNA for analytical purposes, and I thought that it will be good to add on to the facts or the principles from how to collect DNA from saliva. And as you know that. Like all of us know that from saliva, collection of saliva, one of the golden standard is passive rule, but then there are minuses and pluses, so I'm not going into that, but I'll show you an interesting saliva collection device. And before that, I will have to thank Dr. Paul Desmond Sloy, because this device is from OSS Diagnostics USA, and Dr. Paul Desmond Sloy is the CEO and founder of OSS. So basically, he has come up with this device, and which is very, very useful. And at the initial stage of sampling only, you get a purified salivary DNA. And this DNA is very, very stable. You can actually transport it. And I will just show you, I will just give you some tips and tell how this device works. I'll go straight to the thing. So this is how it comes in a packet like this. So this is called as DNA sal. And when you get this device, so I will just open it up, and this is how it works. So let me just open it up. Well, this is not a sterilized packet. This is just yeah, this is not a sterilized, but see. So this is some one vial, which if you're able to see, I'll just hold it. There is some liquid inside, right? So that's the stabilizing solution. So, and if you see, there are some labels here. So this is the stabilizing rinse solution, which actually it is written as stabilizing rinse solution for rinsing only and do not swallow. So basically here we will write the patient's name, and this is the collection tool for collecting the saliva. So what we do is we open up this, and then we ask the patient to take this particular liquid inside the mouth, and then rinse rinse inside the mouth, and not swallow, but throw that again into this. Now, as you know, when we talk about collecting DNA, all right, from saliva or oral cavity, it is not only about saliva, because we have DNA in buccal cells also. So what we do when we have to collect DNA entirely from the oral cavity, then we need to address or take up those cellular DNA also. So there is another provision with this. There is a thing like this, which has some tooth on this, and it has a breakable handle. This handle is breakable. So what we do is we put this inside the mouth and press the cheek from outside like this, and we scrub it, putting it inside, and straight away take it up and open it and put inside and just mix it. And then what we do, once we put this inside, we break this handle. So whatever this tooth like structures that you are seeing in here, the cells will get stuck, and it will, you can actually collect enough buccal cells, and you do not contaminate anywhere, and you just take it and put it inside and break the handle, and you throw off this handle, and again you you just tighten it, and it is ready to store and ready to ship. And the DNA is very, very stable in this, and it is a very safe way to extract pure DNA from saliva. Rather, I should put it as from the oral cavity. So that was a very fascinating product which can be used, and this can be stored at room temperature. It is very stable. DNA doesn't degrade at all. It is as stable as more than a month, because when if you want to ship it somewhere, you can do that. So, and because the solution that I showed you is a stabilizing solution, again it stabilizes the DNA, whatever we have collected, whatever is there present in the saliva, it stabilizes that. Stabilizes as in it doesn't degrade the particular DNA that we intend to analyze. So, well, I don't say that we have to use this particular device in all the cases, but yes, when we are looking for a depending on our downstream analytical work and what we are intending to analyze with salivary DNA, I think there are many advantages with this particular device when we compare it with passive drool, because as you know, passive drool is we will have many impurities, proteins, mucins, all these things will be there. And so I think we got some good insight, and that is all from today. From me today. Nothing more to add on. I think Rajeshri and Udita has beautifully explained the different aspects. And to be very honest, and we just planned it. It was not planned actually, and we thought that okay, it's National DNA Day, and what can we do together? And we were just trying to connect each other. Then we thought, why not to connect with all of you together? So that was the thing that we tried to do, and I hope all of you have enjoyed. And thank you, Udita, and thank you, Rajeshri. And also thank you all the viewers who had been live with us, because it's late night. But I thought that nowadays we are not following that particular schedule. So we generally go and go and do take our brunch. It's all becoming combined and compact. So that is how it is going. So that's all from all of us, and so stay well and stay safe. And I think in some point of time we will all come back in a regular days. So just we need to be calm, and just we need to accept and go ahead. So thank you, Udita. Thank you, Rajeshri, and thank you, everybody. So, and good night. Bye bye.
Bye, ma'am.